Hi everyone. So there have been some questions about how to process all of the information that I covered on organic chemistry. So what I want you to do is get out a piece of paper, just a big full-size blank piece of paper, and fold it into either quarters, so fold it in half and then fold it in half again like this, or you can fold it in half and then fold it in half again like this. So either way, you basically are dividing your piece of paper into 4 sections. I'm going to go through and summarize the highlights of each type of organic molecule and fit it on 1/4 of that piece of paper, so that by the time you're done, you'll have a piece of paper that maybe looks like this and has the highlights of all of the organic molecules. Now keep in mind it's not going to have everything on here, but it's going to at least give you a really good starting place of things to memorize. So the first thing to remember are, what are the 4 types of organic molecules? So we have carbohydrates, lipids, proteins, and nucleic acids. So let's start with the easiest one first. We've got carbs. Now, what are the monomers or building blocks of carbs? The monomer, which means the single part, is going to be a sugar or a monosaccharide. Now, I'm going to use an entire slide for each type of molecule because it's kind of hard with this pen to write very small. So on your piece of paper, you're going to try to keep everything I write on this slide on 1/4 of your sheet of paper. So what are some examples of monosaccharides? We've got glucose, which is important because that's blood sugar. And we had fructose, which you can find in fruit, although you can also make fructose. We can have bigger sugars, which are called disaccharides. So these have 2 sugars linked together. Examples of those included sucrose, which is table sugar, lactose, which is milk sugar, and maltose, which, as we said, you can find in malt, or to make a chocolate malt, or to feed yeast so that you can make beer. And then we have really big sugars called polysaccharides, and examples of those include starch, which is a plant energy storage molecule that you can digest. We have cellulose, which is a plant structure or structural polysaccharide that gives some rigidity to the cell wall of a plant cell. And then we have glycogen, which is animal energy storage. So some important points. Starch is digestible, whereas cellulose is not digestible. So then we talked about why you would need to eat cellulose. If you can't break it down, that means you cannot get energy from it. Well, we use cellulose to help clean out the digestive tract, to make you feel full longer, which helps with calorie control, and to slow the absorption of glucose. So in general, though, for most carbohydrates, the function is going to be energy. So in animals, we use glucose in our blood to help make ATP, and we use glycogen that's stored in liver and muscles to help replenish the glucose as it gets used. All right, so that's carbohydrates. So now let's talk briefly about proteins. So proteins, monomers are amino acids. and there are 20 essential amino acids. The recipes for making proteins are found in the DNA. So these are really diverse molecules because your genes only code for how to make proteins, so you have lots of different kinds. You have amino acids all linked together in a big, long polypeptide chain. And those amino acids are held together by a specific type of covalent bond called a peptide bond. That'll be important because when you get to digestive system, they'll talk about how pepsin in your stomach helps you to break apart those peptide bonds. Now the functions are very diverse because we have lots of different types of proteins. So we broke it down into 7 categories of functions. So we had movement. So these would be things like actin and myosin. We have proteins that regulate metabolism. These would be enzymes like pepsin or lactase. We have structural proteins. These would be things like keratin, which is found in hair and nails. We have proteins that help with transport, like hemoglobin, or albumins, or membrane proteins that do active transport and move stuff across the membrane. We have defensive proteins, like antibodies, that bind to foreign substances to mark them for destruction. We have buffers, and we didn't discuss any specific types of proteins that do that. And then we have proteins that do coordination and control, and these would be protein hormones. Don't forget you can also have steroid hormones. And the example I think that we gave for a protein-based hormone would be things like insulin, or growth hormone is another. And then we talked about enzymes, and they're going to decrease the activation energy of a reaction. And they're going to do that by binding to a substrate with an active site. You can get into that more with your notes. If we denature a protein, then we change its three-dimensional shape, and then that means that our function changes or stops. It can't work if it's changed shape. All right, so that's a really quick summary of proteins. So now let's talk about lipids. Now we talked about there being 3 different main categories for lipids. We have the true fats, steroids, and phospholipids. So let's talk about the true fats first. So these would be things like lard, butter, oils, animal fat. The monomers, or building blocks for true fats, are glycerol and 3 fatty acids. You can have a saturated fat or an unsaturated fat. With a saturated fat, you're going to have a molecule that looks a bit like this. So this is just continuing to the rest of my molecule. We have to make all of these carbons happy by filling them up with hydrogens. So they are saturated with hydrogen, which means this molecule has the max number of hydrogens, and it has no carbon-carbon double bonds. And therefore we end up a solid at room temperature. So this would be things like butter and lard, animal fat. An unsaturated fat then is not saturated with hydrogen, so it does not have the maximum number of hydrogens. So if we kind of draw the same thing here, maybe these are all filled with hydrogen, but we're missing a couple of hydrogens. So to fix that, we stick a double bond between the carbons. That way my carbon still has 4 bonds, and this carbon still has 4 bonds. So we do not have the maximum number of hydrogens. We have carbon-carbon double bonds, and therefore we end up with that kind of crinkly molecule that's all bent so that it doesn't stack nice and tight at room temperature. So we have a liquid at room temperature. So these would be most plant oils. Then we have our function. We can talk about the function of those guys. So our true fats functions are going to include mostly energy storage, specifically long-term energy storage. Longer term than I would like, that's for sure. And we have, of course, insulation and some protection or padding for organs. In particular, there's an important fat pad around the kidneys. All right, so then we have the steroids. And we basically just talked about some examples. Sex hormones, thyroid hormones, and cholesterol are all steroids. Now when you get to cells, that's going to be important because depending on the type of molecule it is, it's going to signal cells differently whether I'm a steroid, which is a lipid, or whether I'm a protein. So that'll come up in particular when we get to endocrine system and we talk about the different types of hormones and how they signal our cells. What are the functions of steroids? Most of them are going to be involved in signaling, although they can also act as carriers, or they can help build the cell membrane, like in the case of cholesterol. And the last category that we have here would be the phospholipids. The monomers for that are going to be glycerol plus now only 2 fatty acids plus a phosphate group. and that phosphate group has some other R group or functional group attached to it. This part of the molecule we think of as the fatty acid tails. These are going to be hydrophobic, whereas this is going to be hydrophilic. And the main function of these is to help regulate what moves in and out of the cell, because it makes up the majority of the cell membrane. So this is the main component of cell membranes. So that helps us to regulate what moves in and out of the cell. Our last category would be the nucleic acids. The monomer for nucleic acids are nucleotides, and nucleotides are made up of a phosphate attached to a sugar and some nitrogenous base. The bases in nucleic acids differ depending on whether you're talking about DNA or RNA. In DNA, we have adenine, thymine, guanine, and cytosine. In RNA, we have adenine, guanine, and cytosine, but instead of thymine, we have uracil. There's a rule for which ones can pair together. We get A and T, G and C. And when we're actually reading DNA and making proteins, we do have to pair RNA up. So we're going to pair A and U and G and C. For DNA, the easy way to remember which ones pair is you can just think that DNA is where it's at. If I were to take a strand of DNA and let's say the bases are A, T, T, G, C, A, C, you should be able to give me the complementary strand that would occur when I'm replicating this DNA. So you're going to use your base pairing rules. So A would pair with T, T with A, C, G, T, and G. So this would be its complementary strand. Now, what if I had DNA, and let's use the same strand, but now I want to make mRNA from it because I'm doing protein synthesis. If I'm going to make a complementary RNA strand, remember, I'm going to use the same complementary pair rulings, but RNA has uracil in it. So it's going to be A pairing with U, and then T with A, C, G, A with U, and then G. So my RNA strand should look very similar to my complementary strand, except that I have U's whenever I'm using RNA. What are the functions of nucleic acids? Well, for now, we kept it simple, and we said that DNA is the recipe for making proteins. And we said that RNA are basically the chefs, so they're going to make the proteins. There's also a special nucleotide that we talked about, which was called ATP, which is a base attached to a sugar, attached to a phosphate, but then we stick 2 extra phosphates on it. And these bonds between the phosphates have lots and lots of energy that we can tap into when we break them. And there's a really good site for helping you kind of visualize the structure of DNA and how it replicates, and that is dnai.org. All right, so that's your quick summary for organic molecules. Hopefully that helps. I would practice this before you get into lots of details on things.